Hey everybody, we're continuing on with from gene to protein, and right now we're going to talk about how that pre-mRNA is modified so that it becomes messenger RNA that can be used by the ribosomes to be translated into an amino acid sequence. So we've got three different types of modifications we're going to talk about. We've got a 5' cap, a poly A tail, and splicing. So a 5' cap, you have a modified guanine nucleotide that's going to be added to the 5' end. So it's going to protect that mRNA on that 5' side from digestive enzymes. It's also a recognition sign for ribosome attachment. Next you have a poly A tail, so poly meaning many, A for adenine, and so this is going to be like 150 to 200 adenine nucleotides that are added to the 3' tail. And so again, this is protecting now the other end of the mRNA from digestive enzymes, and the tail also aids in mRNA transport from the nucleus. So this is how, or it helps get the mRNA out of the nucleus and into the cytoplasm where it can be translated by ribosomes. So, quick comment, the head and tail areas often contain leaders and trailers. So these are going to be areas of the RNA that are not actually read and are not actually translated into an amino acid sequence. So this is an old sentence here from this lecture, but it's similar to the leaders or trailers on cassette tapes. Do you guys know what a cassette tape is? So think of like an old school VCR, maybe like a Disney movie, like the big VHS. If you've ever watched those, you know that at the beginning and at the end of the movie, you're going to see like kind of the fuzzy screen because there's nothing on that tape. And so that section of the tape is sort of there to protect the actual movie part. And so the 5' cap and the poly A tail do the same kind of thing for the mRNA sequence. Next, we've got RNA splicing. And so this is going to be the removal of non-protein coding regions of the RNA. And then you take those regions that code for protein and you splice them back together. So this is kind of what I'm talking about here. We've got our pre-mRNA. So we've already added this 5' cap and the poly A tail. And now we've got these sequences called exons and introns. You can see we kind of alternate between exon, intron, all the way through. The exons are the important part. That's the stuff that we need to keep in the mRNA because that's the part that's actually coding for the proteins. The introns are essentially just extra information that doesn't need to be there. It doesn't code for anything in the protein, and actually if it was left in, it would mess up the protein that's being formed. So the introns need to be cut out, and then the exons need to be spliced together. So introns are called introns. It means intervening sequences. So they are sort of intervening or interrupting that mRNA transcript, and so they need to be removed from the RNA. And then the exons are going to be those expressed sequences of RNA that are then going to be translated into amino acids. So now, how do we do this? How do we cut out the introns and join the exons back together? And we use something called a spliceosome. And this is going to be made of snRNA, and you see this snRNP, all right? So snRNA and SNRPs, okay? So snRNA is small nuclear RNA. It's about 150 nucleotides long, and it's a structural part of the spliceosome. And then the snRNPs or SNRPs are small nuclear ribonucleoproteins. Okay, I know it's a mouthful. And these are made of snRNA and proteins. And so they join with other proteins to form a spliceosome. So here is kind of what we're talking about. We have this pre-mRNA transcript again, so we're kind of zoomed in. So you can see we've got a 5' cap, one exon, an intron, and then a second exon. So now we need to find a way to cut out this intron, but then also attach exon 1 to exon 2. And so what we have here is a protein. So the purple is going to represent protein, and then the snRNA is going to be the looks like a red squiggly line. What that is that's a molecule of RNA. When you have the snRNA with the protein, that's your SNRP. Then you've got other proteins. These are all going to come together to make what's called a spliceosome. And so what is going to happen here is you're going to sort of pull this pre-mRNA transcript here. You're going to pull it together. The spliceosome is going to cut apart the molecule, so it's going to cut out that intron, and then it's going to pull the two exons closer together and bind them. So now you have exon 1 and 2 joined together, and you have the intron that has been removed. So once you do that to all the introns in your pre-mRNA, you end up with an mRNA transcript that has been processed. So now we've got our 5' cap, we have our poly A tail, and now we have only the coding regions of the mRNA. So ribozymes are going to be RNA molecules that act as enzymes, and they're sometimes intron RNA, and they cause splicing without a spliceosome. Okay, so ribozymes are RNA molecules that can act as enzymes, and they are sometimes contained within the intron RNA, and so they can actually cause this splicing to happen without the need of a spliceosome. So why are introns there? What function do they serve? Well, not totally sure. So it could be leftover DNA. It's a way to lengthen the genetic message. It could be DNA from old virus inserts. It's also extra bases that could help to create new proteins. So there's a couple possible functions here. But either way, they need to be removed from the mRNA before you actually make the protein, because it's not coding for anything that's necessary in that protein. So again, we have our final RNA transcript. So we have our 5' cap. We have the coding segment that has all introns removed, and then we have our poly A tail. So your lecture question: why does mRNA need to be processed? How would polypeptides be different if RNA processing didn't occur? All right, thanks guys. Make sure you send me a message with an answer to your lecture question.